Hi, welcome to this exciting conversation on the transformative impact of artificial intelligence in bioinformatics. Today, we're honored to be joined by Dr. Subramaniam, a leading expert whose groundbreaking work bridges computational biology and the life sciences and genomics. With a career dedicated to advancing our understanding of these complex biological systems, Dr. Subramaniam has contributed significantly to areas such as genomics, pathology, and translational research. Bioinformatics and computational biology as a field is at the intersection of biology, computer science, and data analysis. It provides the tools to manage and interpret the ever-growing volume of biological data generated by technologies such as next-generation sequencing, single-cell analyses, and imaging techniques. AI has become a critical driver in this space and many others, helping researchers uncover patterns, predict outcomes, and develop insights that were once beyond reach. In today's discussion, Dr. Subramaniam will guide us through the many ways AI is reshaping the landscape of bioinformatics. We'll begin with foundational questions about the field itself, and then explore pressing challenges such as noisy or sparse data, data privacy, and making sure that these AI models are more interpretable and reproducible in fields such as Alzheimer's, metabolomics, and breast cancer, highlighting its role in early detection, biomarker discovery, and treatment optimization. Finally, we'll look towards the future, discussing emerging trends, ethical considerations, and cross-domain applications that could define the next generation of AI-driven breakthroughs in science and medicine. Dr. Subramaniam, thank you for speaking with us today. It's a real pleasure to have you on the show.
Good morning, Srijan. Thank you so much for interviewing me.
Yeah, so today's focus, as I just mentioned, is largely about AI and how it's set to transform human disease as a whole and our understanding of it. So to start us off, do you think you can tell us a little bit more about your current research and what you're doing?
Yeah, thank you very much. Let me give you a brief preamble to how biology has transformed into data science so that it'll lay the foundation for what we do in my laboratory and also in laboratories across the world. If you cycle the clock back to 50 years ago, biology grew as a very experimental and empirical science where people did extensive amount of experiments, following which they derived mechanisms based on those experiments, which led to further exciting discoveries in biology and in life sciences. Several things that transformed the landscape of biology, starting with the discovery of DNA and subsequently understanding the blueprint of life, how they showed the central dogma, which means that the DNA produces DNA, which is the blueprint of all of living systems, forms RNA, which then forms proteins, which are the workhorse of cells and produce function and, consequent phenotypes, which lead to normal and pathophysiology. And so this, once this conscious thought emerged, two revolutions really transformed biology at that point. The first was the genomic sequencing of the human genome. The sequencing of the human genome started on a scale where people felt that it would take decades, if not, half a century to sequence the human genome. However, the whole landscape changed due to dramatic advances in technology, which gave rise to what's called the shotgun sequencing approach, and which led to within 5 years, the entire human genome was sequenced by a number of people. And this was the foundation for the body of knowledge that started emerging in terms of large data. The genome is only the starting point. This was followed by dramatic changes in other technologies, such as high throughput next generation sequencing methods, which gave rise to RNA sequencing, transcriptomics, single cell transcriptomics, and spatial transcriptomics, and all these methods which were really gauging living systems in excruciating detail, which provided the framework for deciphering the mechanisms that lead to function in biology. However, this came, and this was also accompanied, I should add, this was also accompanied by other omics methods, such as proteomics, where people measured proteins in a large scale in cells and living systems, tissues, metabolomics, which is really now taking off in a huge way. And all of this gave rise to the birth of big data in life sciences, which was presciently dubbed by the Nobel laureate Wally Gilbert. I mean, in 1980s, so as early as 1980s, he declared that biological sciences will soon become a data science. And so he was prescient in his thinking about where we are going. However, this big data, I mean, measurements and revolution in biology could not have happened without machine learning, deep learning, and now the modern AI. And as I'll walk you through a number of examples from the research that we do in my laboratory and in other laboratories, I'll point to the fact how AI is transforming the way we derive knowledge in biology. And towards understanding this, I want to add one more thing before I go into specific questions, which is that unlike data-driven sciences in physical sciences or in other computational sciences where data is well ontologized, where you have finite elements of data. I mean, if you take, for example, data in the financial market, you're going to have currencies, you're going to have exchange rates, you're going to have defined terminology, which is universally accepted, which makes it easier to do computing with data. However, biology, having grown as an empirical science, has tremendous amount of complexity associated with ontology, which means definition of terms and the relationships, objects and the relationships. And so this complicates learning using data methods, using learning methods, makes it much more difficult. However, with modern AI, with generative AI, this is really becoming easier, as I'll illustrate to you as we go along. And this is where the holy grail of the future is coming into picture in biology and life sciences, where we believe that generative AI and LLM has the potential to transform our ability to translate data from large-scale measurements into knowledge that can in turn be applied towards solutions in pathology. So this is where we are headed towards, and I'm happy to answer questions on specific issues with any topics that you're interested in discussing.
Uh-huh. Yeah. So you mentioned LLMs and kind of integrating multimodal data of any of kinds. Now, a kind of an issue with the way we handle that data, as you mentioned, is that biological data is very interconnected, and another issue is that it's not exactly discrete. And one thing that people have been noticing is that when they actually tokenize the data with existing methods, it leads to very faulty analysis downstream because that inherent interconnectedness is not only lost, but also a lot of the information is lost. So, what are your thoughts on handling that?
Yeah, I mean, you allude to your question really connects a large domain of life science data as we come across. Number one, you're referring to the fact that the heterogeneity and the interconnectedness of data measurements in living systems makes it more complex in terms of how to deconvolute at the first level data, even prior to introducing it into a learning system. This is what I refer to briefly as an ontological consideration, where, I mean, terminology, terms, I mean, use of, connection, use of meaning of words, how they are linked to each other. This is really it's like a cottage industry. It's grown in a heterogeneous manner without having a structured vocabulary which would help you connect all these things to feed into a learning system. So this is the first level of complexity that makes it daunting for a learning system. That's number one. Number two, the fact that time scales and length scales, especially let's talk about length scales first. Length scales in living systems range from, 10 to 15 orders of magnitude. If you consider a single, molecule, which is essentially a nanometers wide, or if you consider a human size, which is 2 meters long, and if you consider, in fact, an ecosystem, which is much, much larger, you can already see that the length scale becomes very large. And how do we really deconvolute this length scale and look at the interconnectedness between different scales? And this is a very, very complex issue. The third point, which you alluded to, deals with time and causality. Unlike in measurements where we learn from systems, like even in discretized systems such as climate, for example, if I'm going to measure, or the planetary system or the space system, if I'm going to measure, make an observation on climate, I'm going to make it at a given time. And I can be reasonably certain that if I'm going to make these, continuous measurements over a period of time, I can use the equations to extrapolate the behavior of the system, which is what we do when we predict hurricane paths, for example. And so it's feasible to do it in systems which are well behaved, or even if they are not well behaved, if they have certain modalities or patterns which can be inferred from a learning system. However, in biological systems, causality, which is measured in longitudinally using time series data, is not always very clear. And this is what makes it very complex. If you make a one-time point measurement or a snapshot measurement of data, it doesn't tell you anything about the system. I'm going to give you an example. If you take a measurement through your iWatch or a Fitbit, if you're measuring longitudinally your blood pressure or your heart rate, it doesn't predict a priori a sudden change in heart rate, which gives rise to tachycardia or ischemia. It doesn't tell you anything about it, unless you have other events that in addition you're measuring, which are in turn predicting or postulating a causal connection between these events. So this is what makes biological measured data very complex. So you ask the question, where are we going towards in large language models or in generative AI towards addressing these issues? Towards addressing these issues, number one, we need to have a large body of data that needs to be measured. And this is really happening as we speak. In fact, as you know, from your own experience, there are, people have transcriptomic data with the measurement of 20,000 or 30,000 genes across 50 million cells. This now starts to become respectable, where people are beginning to understand how to look at cell heterogeneity using learning models. Or for that matter, now, as you know from the UK Biobank project and projects across the world, now millions of people are being genome sequenced. And so once we reach the point of about 1 billion people genome sequenced, which is about an eighth of the population of the world, we will be able to start predicting meaningful things in terms of connection between genotype and phenotype. And so these are types of emergent future that we are thinking about in going towards LLMs and generative AI. I didn't touch upon the details of the LLMs and generative AI. We can go forward when we talk about specific examples with those.
Sounds good. So with the data that's being produced right now, a major issue that I think all of us can agree upon is the data itself may be more homogeneous than we would like. And what I mean by that is the UK, sorry, I forgot the Biobank, the data that they're collecting may be inherently biased to Caucasian men or women. And because of that, how do you say that we handle the inherent data sparsity that we get in certain groups of people if we were to apply the learnings from this data in the future to all people?
This is an extraordinarily interesting and important point. Most measurements, if you look at large scale measurements, these are expensive measurements. They are not, measurements which are economically viable. As a consequence, they reach only the strata of society which are in the top percentile of financial stability and wealth. As a consequence, if you take, and also it's geographically nuanced. If you take, for example, the largest amounts of human genome sequencing, it happens in the West, especially in Europe, in UK and in US, North America and US, where we have large scale sequencing that's going on. If you look at other measurements, other high throughput measurements which are expensive, these are also happening in ethnic populations which are unique to geographic locations and which are based on financial considerations. And this introduces a significant degree of complexity. If you look at the UK Biobank project, 99% of people who are sequenced are white or Caucasians. And as a consequence of that, recognizing this is an important point, a number of countries in the world and even United States, we are starting to think about ancestrally diverse populations. In fact, the NIH has launched a project on ancestrally diverse populations where they are looking at specific subpopulation level data. For instance, they're looking at Native American um, pockets in the country where you could do extensive amount of next generation sequencing. And the same thing is true in, for example, in other parts of the world. China has launched a very large sequencing project. India has launched a Genome India project. And there are South Africa, in Africa, for example, NIH has launched a Genome Africa project. So these are all in long-term vision in the coming decades, likely to provide more data that will enrich our understanding of population specific by the eliminate some of the biases that we are looking at. Having said that, I do want to point out that we are very, very largely similar to each other in terms of our genotype. And even though there are specific, ethnic population specific differences, by and large, we are very similar. In fact, my colleague, who is program director at NIH, he's often, he quotes or he uses the data, genome data he has at the moment to say that there's more diversity within African subpopulations than between Africa and the West. And so this is really an important point to keep in mind that despite the fact that we have, we don't address ethnic diversity, we don't address gender for that matter. In fact, the majority of the sequencing is done in Western population more in male than in female. So despite these issues, we believe that there's going to be a lot learned. We will fine tune the technologies, we'll fine tune the methods, we will look at what is being common. And so this will help us go towards the next generation future for addressing the universe at large.
Yeah, that's honestly very great. Understanding not only on a smaller level, but on a larger level. With these large language models, what we're able to do is really model the connections between these different parts of each genome that everyone has. So I have a question more specific this time, particularly about breast cancer, which I know you do work quite a lot on. So with the growth of the application of AI, how do you suggest that we really take advantage of machine learning and AI for not only assessment of breast cancer using mammograms or maybe blood tests in the future, but also prognostication and possible early detection? Yeah.
It's a good question. Let me address this issue. If you again turn the clock back 50 years or so, a sizable population of women who got breast cancer, especially if they were not detected very early, simply had a high mortality rate. I mean, they died. And this is a consequence of the fact that our measurement modalities, detection modalities were primitive, and our treatment modalities were primitive. And so how do we address this issue? So this is what started National Cancer Institute several decades ago to launch initiatives towards overcoming cancer across diverse population and focused it, in the focus that you're referring to, especially in breast cancer in women, was a large initiative. This was followed by a number of national initiatives. As you know, the Moonshot Project, initiated by President Joe Biden, in fact, addressed this issue of curing cancer at some decent level. And this also addressed the breast cancer issue. Now, let me focus on exactly the breast cancer issue. Women are susceptible to breast cancer because in physiology in women, birth and post-birth, as well as over lifetime, hormonal changes introduce significant changes in remodeling of the mammary tissue and the breast tissue. As a consequence, there is a higher chance, higher probability of the normal tissue mutating into a tumor tissue. This is guided by a number of factors, including genetic factors, environmental risk factors, including diet, including pollution, including other factors like socioeconomic status. So these factors contribute to breast cancer. However, detection of breast cancer used to be done for several decades using mammography, where people were looking at both imaging and simple methods of looking for lumps in women's breast and so forth. And this was a standard practice for a long, long period of time. And then in the worst case, when they felt that there was a potential for breast cancer, they would do mastectomy, where they would remove the breast. This was used to be the common practice for a long time. Then came the drugs, which were included chemotherapy and radiotherapy drugs, which would essentially kill cells, both normal and cancer cells, hopefully more cancer cells. And this resulted in better prognosis for women. Then came the time when we could take the tissue and either do imaging of the tissue in such exquisite detail as to trace the anatomical origins of where the tumor begins or where the tumor is spreading, and as well as extract tissue to do multi-omics measurements, which will tell us about the tumor cells, the tumor niche, the environment, which will contribute to the growth, and all of that in exquisite detail. However, these vast amounts of data need to be processed to understand, A, the mechanistic details of how tumor begins, progresses, and metastasizes. Two, to understand what types of treatments would be appropriate for what types of conditions and what women. And three, look diversely across epidemiological status, across population of women, to look at are there nuances specific, for example, to African women of African descent or European descent or some other descent. So these are all factors where large data, large imaging data, and large omics data can provide substantial insights. This is the focus of my laboratory over the last several years in deciphering what is happening at the molecular level, to the tumor, and are there potential interventions which would help cure the cancer. So first, we asked the question, would we be able to subtype this tumor so that we can ask the question, one treatment does not fit all. What's good for the goose is not good for the gander. So therefore, we want to have a modality which is specific to a type of breast cancer. So we did subtyping of breast cancer. In fact, much prior to our work, decades before our work, there was subtyping of tumor based on whether it is estrogen receptor, hormone receptor positive or hormone receptor negative. And women were diagnosed and then appropriately given treatment, hormone treatments or hormone therapy treatments, or if not, if they were negative, they were given other treatments. So this was a common practice even before. However, we wanted to bring this into a modern science. So we first used the omics measurements to look at learning methods. First, we used AI methods, such as auto-encoded learning methods, to identify markers, which would help us identify which type of tumor, which type of breast cancer is the woman susceptible to, or is currently, exposed to, or has. And so using this, we published a paper, which is now under review, on auto-encoded methods for subtyping tumor with specific markers. We then asked the question, how do we take all this data, and can we do use topological learning methods to look at topological analysis of data? And just recently, we have a paper that's being published which shows how topological data analysis has the potential not only to subtype women tumor, but also predict a new subtype of tumor, which is amenable to a given type. This is forthcoming, and we are very excited about the ability to look at large data and do topological analysis of this data. This uses the idea that shape of data and topology of data is very important. Now, third, when we focused on one type of tumor, triple negative breast cancer, we are able to ask the question, are there subtypes within the triple negative breast cancer which would be useful for predicting, postulating treatment? And this was motivated by the fact that today it is common practice to do immune therapy for most tumors or checkpoint immune therapy, and only 30% of women respond to immune therapy, and the others not only not respond, but there are adverse effects arising from immune therapy. So we want to predict a priori, can we really have a subtyping, which will tell us which women should be subject to immune therapy. So using large transcriptomic data, we're able to predict subtyping. We're able to subtype these women to identify classes of women who would be amenable to checkpoint therapy and others who require different forms of therapy. We then decided to do a digital landscaping of the tumor tissue and the surroundings. And this digital landscaping provided spectacular amount of details in terms of regions which are invasive regions of tumor, which are adjacent regions of tumor, even suggesting to us how early tumor transforms, or early cells which are not completely tumor and transform into tumor cells. And so this type of prediction was only possible using extensive amount of omics measurements, followed by learning methods of various kinds. And in fact, we are now using deep learning methods to identify specific markers which will provide an idea of good prognosis versus poor prognosis in tumor in women. We anticipate this to be the future, where such measurements are going to be made on not just breast cancer, breast tumor tissue, but all tumor tissue, where we'll be able to predict, prognosticate not only the type of disease, the potential modalities of treatment, but also suggest the lifespan and suggest the lifestyle requirements that should be adjusted to go towards the future. So this is what we are hoping would be the holy grail for treating cancer.
Yeah, and with the improvement and increase in the amount of data, slowly but surely we'll be able to have enough information regarding previous cancers to be able to make that decision. So you said something about the topological analysis. I had a quick follow-up on that. So when you refer to topological analysis, are you referring to analysis between different scales as we talked about at the beginning? Or do you mean in a single level and analyzing its outcome?
In theory, you could do it across different scales, although the work that I'm describing to you is across the transcriptomic scale. So we took transcriptomic measurements from the Cancer Genome Atlas for women with different types of breast cancer. And we agnostically, without providing any information on what type of breast cancer, we used a topological data analysis, use the shape of the transcriptomic data to decide whether it can topologically partition the data into different subtypes. And we were really absolutely stunned to see that it was not only able to partition the data into different subtypes, but also partition one subtype further into two subtypes, which gave us insights into treatment. And then we went back to scientific literature, clinical science literature, to show that some women in this particular modality of disease did not respond to treatments, which gave us great confidence that this topological analysis was able to discern a subtype which was not previously discovered.
Yeah, that's brilliant. I mean, to see AI is able to not only make connections that are already existing, but actually provide further insight is very amazing. So regarding analysis of multiple levels of breast tissue, I mean, just for context for the viewers in case they didn't understand, I think what we're referring to with the different levels is at the top, maybe something like a mammogram where the scale is in centimeters, and at the bottom, maybe omics data and more very fine imaging that's in the micrometers, really bridging the gap between the two to do an overall comprehensive analysis. So, with this, were there any promising insights in that bridge between the two that you found to be up and coming?
This is a really, really good question. So let me elaborate a little bit on your question before answering your question. At the highest level, we have millimeter level imaging using MRI or other methods of imaging. We can get millimeter scale resolution where you can possibly see presence of tumor, presence of lumps, presence of tissue. We don't know whether it is benign or, malign tumor, but however, you can detect at the imaging level, large scale, provided the size is grown to a significant amount. You're referring to imaging at that level. Now, when you biopsy a tissue, when you do either a needle aspiration or a core biopsy of a tissue, you can now look at the tissue at a landscape which is more fine-tuned, which is at the level of about microns or tens to hundreds of microns, which will tell us about in a more detailed and nuanced way, the tissue and its microenvironment, and perhaps tell you also whether it is metastasizing or not to some extent. So it will provide you with the next level understanding of the tissue in detail. If you now take the same tissue and profile it using digital spatial transcriptomics, it now gives you at the cell level understanding, tells you cell heterogeneity in the tissue. Is it a normal tissue? Is it an abnormal epithelial cell that's housing this tissue? So we'll be able to start looking into finer levels of detail. Then you go to the next level of detail using the transcriptome. You start looking at which genes are expressed. Are these genes aberrant genes which alter normal functional pathways in a breast epithelial cell and its microenvironment? And so this is the building of the scales or bridging of the scales. And we are at very early stages in terms of doing this digital pathology. I call this digital pathology. We are at very early stages. In fact, integration of all these scales is where the future is going. This is where I'm looking forward to generative AI and LLM providing us with much more information and insight into bridging the gap between length scales and time scales.
Yeah, and it's going to be interesting to see how data collection and measurement techniques improve to allow for the efficient and cost efficient methods of taking it over a very short periods of time so that we can generate that time scale data as well. Yeah, so you talked about on the transcriptomic level, understanding gene functions as part of analyzing and possibly producing treatments for diseases. So how does AI really support the prediction of gene function for certain uncharacterized genes that we are not yet fully aware of or not yet fully understanding of?
This is also a very, very interesting question. Clearly, genes are only a microcosm of the big picture of the functioning of a cell, a tissue, and a human being or a living system. It's only a microcosm. While genes do play an important role, gene function, gene expression is followed by translation into proteins, which are the workhorses of the cell. These proteins are not correlated 100% with genes. You may have expression of a gene, but you may have no protein. Conversely, a protein lifetime may be long enough that you don't need to express a gene. The gene may not be expressed during the lifetime of the protein. And further, proteins have function. They act as enzymes, they act as transporters, they act as transcription factors, they perform multiple functions. For example, proteins are involved as enzymes, they function as enzymes in metabolizing the food you intake. The food you intake is processed by your enzymes and cells into metabolites, and these metabolites in turn are the ones that provide the source of energy, like such as in mitochondria. So to understand the functioning of living systems, the transcriptome and the genome, the genes, are only a small portion of the large-scale measurements that we do of the metabolome, the proteome, and other types of omes and phenotypic measurements, which we call as phenome. So towards understanding all of this function, we need a systems level approach. In fact, I call my laboratory as a systems biology and systems medicine laboratory because we focus not on the genome, not on the transcriptome, not on the metabolome, but we focus on the integrative function of all these measurements. And this is where AI plays an even more interesting and important role because AI has the power, generative AI, LLM, has the power to find these connections across this data, which is not transparent or visible to the human eye or to the human mind or computable by the human mind. And so this is where AI comes into picture. I will give you a few examples before I go on. Number one, the reason I mentioned the metabolome is because the metabolome is something that's a dynamic quantity, unlike the original genome, which is more or less a static quantity. You're born with the genes that you have. Yes, the genes do mutate, but the mutations are small and we don't detect each mutation in a different tissues is different, so we are not able to measure them very easily. The metabolome is a dynamic entity. For example, if you're doing an exercise, you are going to convert your glucose into, imagine you're a sprinter, you're going to convert your glucose into ATP molecules very quickly using a process called glycolysis, which will help you produce energy instantaneously. If you're a long distance runner, you want to have train yourself to have oxidative machinery function to a longer period of time to produce higher amounts of ATP over a sustained period of time. So metabolomics gives you some information about the dynamic state of human physiology. And the great power of metabolomics is also that you can measure it in body fluids. You can measure it in blood and serum and saliva and urine and feces, which are easily accessible relatively without deep invasive measurements. And so the metabolome can provide a lens into a dynamic state of a human being. We call this as metabotyping humans, just like genotyping, except that the metabotyping is a dynamic entity. Now, let me bring you back to AI. When we now have the technology to measure tens of thousands of metabolites from body fluids, and these measurements can be carried out longitudinally. I can, for example, take saliva or urine over continuous periods of time and do these measurements. And we can investigate our feces. We can investigate, for example, the changes in the microbiome in the colon with the feces. So these are examples of where omics gives you large amounts of longitudinal measurement data. And then where AI comes into picture is twofold. Number one, this data is very vast and large. And to analyze this data, we need to have methods which are, much more sophisticated than simple statistical methods, which only provide correlation and don't provide any causality. Second, if you want to calculate factors which are really feature space, which are going to be significant for predicting the state of the human going forward so that we can have intervention strategies, then we need to have AI to tell us about learning based on longitudinal measurements on the evolution of the system towards normal physiology versus pathology. And so this is where we believe that the integrative omics and systems biology connected to AI, deep learning, and generative AI are going in the future.
Yeah, I think what we're from honestly all the questions in the interview, the real impact of AI comes into the growth of data. With this increased information, there's not as much that a human can do with it as an AI can. The AI is able to analyze A and C and everything in between, and then come up with more complex analyses. So that really brings me to my last question about kind of the black box that's around AI right now. For a biological analysis, there's a lot of information we need to derive. But as of right now, AI and machine learning methods are very close in what the computer can actually learn, which is where a lot of the mistrust and a lot of just the lack of understanding arises from. So from a biological perspective, what is it that we'd like to see from machine learning that might allow us to better understand and better use it in the future for this multimodal data from metabolomics to transcriptomics to imaging?
Yeah, thank you for this question. This is also a very important question, which comes to the very core of AI. How did AI begin? AI began because people felt that we want to have an exobrain that calculates, uses the sensory inputs that we use in our brain, but in terms of digital data, towards transforming it into knowledge. If you think, let's focus first on the human brain. I'm really glad you brought this up because we also work on human brain and neurodegenerative diseases, which talk about how the brain functions. Now, if you think about cognition, the way a brain is an analog computer. So we don't always understand how, for example, a perception of a vision, for example, is translated in the brain into an image followed by an action that we pursue, driven by motor function. And so this is of how we think about the brain, and we have made tremendous advances in terms of mapping the brain to regions in the brain which function, but we still do not understand exactly how the brain transforms the input into knowledge. When we go now to a computer, in fact, von Neumann, before he died, gave a beautiful set of lectures called The Computer and the Brain, which I recommend to your audience to read. They're called the Silliman Memorial Lectures, which provide insights into how the brain and computer are connected. So if you look at now the computational aspect of the brain, what does the computer do? All the computer does is take the input set of features that you're providing, input data that you're providing, transforms it or looks at connections between those using weights for different input data, converts them into hidden features which we do not recognize. They are not human readable features. And these features are fine-tuned to always produce ground truth. At the end of the day, the ground truth is mapped and you train the system. Once you train the system, you trust the system well enough to produce ground truth. It is like a child learning a language so that eventually the child is trusted enough to interpret the language towards taking actions. So the black box that we are talking about is still largely a black box because the features that we are looking at through the intermediate states of the AI system are not clearly interpretable features. Now let's ask the following question. Can I map the intermediate state of an AI engine into something that is interpretable in terms of my knowledge system, my knowledge, basis? If I can do that, then that is starting to become useful because I can also, not only I can understand what the machine is doing, but I can guide the machine towards better learning in terms of biasing or orienting it towards learning. And this is now becoming a very strong feature of modern AI, modern deep learning methods, where we are starting to produce interpretable AI, where we are looking at the intermediate states of these learning system to look at what these states mean. And so this is where we are going towards various methods. For example, the autoencoder, decoder methods really think about this idea of compressing these intermediate states to asking what do they do at different levels. And so it's more than just saying that if I use these weights and these features at this intermediate state, I'll produce this accuracy. That's only a marginal interest. The more interesting thing is whether I can interpret this intermediate state. How much can I go towards the interpretation of how much can I mimic the brain in this black box AI system? For example, my brain has intermediate states in going towards vision or perception, and these are being mapped now very beautifully by using EEGs and other modalities, electrophysiological measurements. And so can I do something equivalent computationally in the AI in terms of intermediate states? So this is where the future is going. To answer your specific question, where are we? What are we looking towards? We are looking towards both trying to go deeper into these learning systems in terms of their states. Don't forget, these are all evolving through states, as well as providing interpretation of the states, and guidance of these states. This is where also LLMs can help us in terms of trying to provide understanding of the intermediate states.
And especially multimodal LLMs. So not only it has an understanding of the data itself, but also maybe natural language so that we kind of understand, I
Exactly.
mean, whatever it believes to be what it's thinking.
Exactly.
Yeah, so that's really all I had for today. Thank you so much for our viewers for watching. Let us know in the comments if there's anything more that you'd like to see.
Thank you so much, Rijan.